he got everything done he wanted to. It was very nice, though. Had a very nice time, and it's always good. I had a few people from this class, actually, who came to Thanksgiving at my house, and we had a good time, so that's always fun, too. But I think most importantly, it was full of food, and it was full of sleeping in late, and whatever, so that was good. Well, the end is near. We are in our 10th week, and I just realized, I promised I was going to bring index cards today, and I just realized I didn't do that. So if you want to come by my office and pick them up, I will give them out starting today, and I promise I will bring them on Wednesday. I just spaced out. I should have had them here today. Okay? As I noted earlier, you do have to get the note cards from me, and if you come to the thing without a note card, then that can be a problem, and the note card has to come from me. So make sure that you get a note card from me. Very important matters. You will have to turn the note card in at the end of the exam, and that's that. We'll say more about that. The exams were made available on Wednesday. For those of you who are not here on Wednesday, you can pick those up. As I noted in my email out this weekend, you do have 3 school days to get corrections and so forth taken care of. Uh-oh. Well, we're in trouble today. Stuck with this one. So I'll probably... Oh, boy. Oh, no wonder. Duh. If you turn it off, it ain't going to work. It'll start a fire? Here's a useful piece of information. If you take a 9-volt battery and you touch it to a piece of steel wool, it will catch on fire. That's useful. That's very useful. Okay, so we do have juice. All right, sorry about that. All right, now we are starting to turn our attention to the final exam. And the final exam, I have decided, is going to be about 150 points. So it'll be longer than a regular exam, but it won't be twice as long as a regular exam. The format will be generally the same, but I may be modifying a little bit to make it probably easier for you to take and shorter to grade. That's 2 considerations, especially in a final exam. So I'll let you know more about that as we get closer to it. I will schedule a review session for the final exam, and that will probably be over the weekend. Haven't decided for sure on that, but probably over the weekend, and I will let you know about that. In your preparation for the final exam, my guideline is, as I mentioned in the lecture on Wednesday, I recommend that you use the 2 exams that you've had so far as a guide to your studying, okay? A guide to your studying. So they make a good outline, I think, of things to study. You're going to see questions related to things that are there. I am, as always, not going to go back and pick out obscure facts about something that I haven't talked about on the final exam. You're not going to see the same questions on the final exam. Occasionally, I throw 1 or 2 in that are the same. But for the most part, you're going to see, questions in that same general topic areas that you've already seen before. Okay? All right. So if you have questions or anything about the process or the exams or whatever, please let me know. And I will be happy to help you, work with you. Well, we are on our last topic now. And that last topic is that of glycogen metabolism. So we've gone all the way through glucose, both the breakdown and the synthesis of glucose. And I hope that I left you with the understanding there's a lot of complexity to it, but that complexity has a logic to it. And what you're going to see as I talk about glycogen metabolism is that glycogen metabolism is very simple at one level. We're going to cover glycogen synthesis and glycogen breakdown very, very simply. You're going to see a couple of enzymes involved, and that's about it. Okay? The complexity of glycogen metabolism is in the regulation. And again, there's a logic to it. And if you understand that logic, you can figure out virtually anything that somebody can throw at you with glycogen metabolism regulation. Now, I'm not going to focus so much on regulation at the moment. What I intend to focus on to start with is to reintroduce glycogen to you, say a bit about the enzymes involved and some considerations with the enzymes involved, and a little bit about how structure of glycogen relates to function of glycogen. So if we look at glycogen, we can actually see glycogen granules inside of cells. Okay, so this tells us that glycogen is a pretty big molecule in order for us to be able to visualize them. So glycogen can contain thousands of glucose residues, and they're arranged in a very branched format. Okay, a very branched format. So I've shown you a picture of the format of the structure before, and I'll remind you of that in a minute. But before I do that, I want to show you a little bit about how glycogen is really essential for being a source of glucose and a place to store glucose. If glucose is a poison and glycogen is not a poison, then we have a place to store a poison and let it out in small amounts so that it's not poisonous. Okay? That's very useful. The liver is involved in that process intimately. If we look at glycogen, as shown at the top, we discovered that glycogen can be broken down by an enzyme called Glycogenphosphorylase that I will say a lot about in just a little bit. And that produces a product known as glucose 1-phosphate. Okay, you've seen glucose 1-phosphate before. Where did you see glucose 1-phosphate before? You may remember? Or has the turkey gone to your brains? Galactose metabolism. Okay, so that's where you saw glucose 1-phosphate. And at the time, I pointed out that glucose 1-phosphate could readily be converted into glucose 6-phosphate. Okay, now notice the name of the enzyme that does that: phosphoglucomutase. What do mutase enzymes do? How do they work? Anybody remember? They have an unusual mechanism by which they work. Okay, I'll give you a hint. Do you remember 2,3-BPG? That was created as a byproduct of the action of a mutase enzyme. So mutase enzymes work not by moving the phosphate from one side to the other, but rather by putting the phosphate on the other end and then removing it from the first one. All right? So that means that during the synthesis of glucose 6-phosphate, there actually is an intermediate known as glucose 1,6-bisphosphate. And just like 2,3-BPG, it can be released by the enzyme, and you can find it in cells. Now, unlike 2,3-BPG, it doesn't have any specific function in terms of telling hemoglobin that metabolism is going on or any of that. Okay? But glucose 1,6-bisphosphate is produced as a byproduct of this reaction right here. Well, glucose 6-phosphate is really the key, because glucose 6-phosphate, you've already seen how it can go and be broken down in glycolysis. You can also see how that glucose 6-phosphate can be converted into glucose. That's the last step in gluconeogenesis, and that can happen in the liver. So it can be exported to the blood. You've seen glycolysis, of course. And there's another pathway that we don't really talk much about here. I'll say a little bit about it next term. And that's the pentose phosphate pathway. Glucose 6-phosphate is a starting point for the pentose phosphate pathway. And the pentose phosphate pathway is a very important pathway for cells. We don't really cover it an awful lot in biochemistry. And that's unfortunate, because it's this pathway that gives us two things that we really, really need. One is ribose. Ribose is necessary for making nucleotides. The other thing that we get out of this pathway that we need is NADPH. And we haven't said really anything about NADPH yet. We've said a lot about NADH. But NADPH is just like NADH, except for it has a phosphate on it. And it turns out that NADPH is used for biosynthetic reactions. We don't really use much NADH for biosynthetic reactions. Most of the biosynthetic reactions use NADPH. So the pentose phosphate pathway is a source of both of these important molecules. So what you're seeing now is glycogen is feeding into an awful lot of pathways that are important for different cells in the body to do different things. Now, what I want to do now is show you the reaction that glycogen phosphorylase catalyzed. So glycogen phosphorylase, remember, catalyzed that first reaction that we saw in the breakdown of glycogen. If we look on the left, we see glycogen. And remember the structure of glycogen. It has glucoses arranged in a polymer of alpha-1,4 linkages. And about every 10 residues or so, it has a branch of 1,6. Okay? Glycogen phosphorylase acts on the alpha-1,4 linkages. It won't touch a 1,6. In fact, it won't even get close to a 1,6, as we shall see. What it does is an interesting reaction. Okay? We've talked about hydrolysis reactions. When we talked about hydrolysis reactions, a good example of a hydrolysis reaction was that which a protease catalyzed. A hydrolysis reaction uses water to break a bond. So, proteases, like chymotrypsin, add water to a peptide bond to break it. They're using water to break it. That's a hydrolysis reaction. This enzyme catalyzes what's called a phosphorylases reaction. It's related, okay? But instead of using water, it's using phosphate to break a bond. It's using phosphate to break a bond. So the product of this is a break between the two glucose residues that were joined by an alpha-1,4 linkage, okay, and the addition of a phosphate to position number 1. Okay? So there's the glycogen that's got one less residue on it. There's the glucose 1-phosphate. Now, this has a very useful consequence for the cell. When we started with glycolysis, if you recall, we went in the hexokinase reaction, we added glucose and ATP together, and the product was glucose 6-phosphate, correct? Now, I've just shown you a reaction where we've put a phosphate onto a glucose, and we did not, in fact, use ATP. My first question is, how is this possible? How do we put that phosphate onto that glucose without using ATP? You've already seen in the case of the hexokinase reaction that there was, in fact, a need to use ATP. How does this enzyme get away with it? Jenny?
Is the energy stored in the glycosylated bond?
Very good. So you're using the energy that's stored in this bond here to facilitate the addition of phosphate. Now, by using the energy of this bond, okay, we are therefore able to make a glucose intermediate with a phosphate on it without wasting an ATP. That's very useful. Because if we had simply used water to break this bond, we would have lost the energy, and then we would have had to add more energy to get a phosphate onto the glucose. This saves the cell energy. Okay? Now, I want you to remember that this glycosidic bond that's between the alpha-1,4, that glycosidic bond has energy. That means if we're going to make that bond, when we go to synthesize glycogen, we're going to have to supply energy in order to make it. Okay? Everybody got that? Okay. So that's a phosphorylasis reaction. Now, as I said, glycogen phosphorylase is an enzyme that is, it not only won't work on alpha-1,6 bonds, it won't work if it gets close to an alpha-1,6 bond. Okay? Now, a lot of people spend a lot of time worrying about the exact numbers of glucoses away that is, etc., and it's pretty straightforward, but we're not going to focus our attention on that. Instead, we're going to note that as glycogen phosphorylase works, it starts at the end of a glycogen molecule, and it works its way inwards. So glycogen phosphorylase will take this template above, and it says, okay, here's a bunch of glucoses that I can nip off of this guy. Here's a bunch of glucoses I can nip off of this guy. And now I've gotten to within 4 residues of a 1,6, and I'm not working anymore. Okay? So that 1,6 bond is poison for the enzyme. It won't go any closer to that branch than 4 glucose residues away. In the meantime, it's produced, in this case, 8 glucose 1-phosphate molecules. That means, therefore, that there has to be an enzyme activity that allows the cell to use these in some way. Otherwise, all your glycogen would stop the breakdown process once you got 4 away from a joint. And there's a lot of different joints, or junctions, or whatever you want to call them. There's an awful lot of those joints, so if you do that, then you're going to throw away an awful lot of glucose that would be useful. So it turns out cells have an enzyme that has a mouthful of a name that we're going to call debranching enzyme. Okay? Its name should say something about its function. It's called debranching enzyme. And debranching enzyme has 2 catalytic activities. And it's a very unusual enzyme in this respect. It has two different catalytic activities. One of them is transferring part of this top to the next, extending the chain here. And the other part, the other activity, is clipping off this last glucose. Okay? Now, so what's it doing? So these two things here, called the transferase and the alpha-1,6 glucosidase, are both activities of the debranching enzyme. So the last two reactions are both catalyzed by the same enzyme that we're going to call debranching enzyme. The first thing that the enzyme does is it says, okay, we are at a branch because we're 4 away from a branch. And it grabs 3 of those guys and puts it on the end of the other one. That leaves behind 1 that it won't transfer. And in fact, it clips that off using water to release free glucose. This is the only place in glycogen metabolism where free glucose is directly released. Why do you suppose it works that way? Why do you suppose it takes 3 and transfers it, and it doesn't take all 4 and transfer it over? There's a reason. What's that? Well, there are different types of bonds, yes. That's not really the reason. Because it's taking a 1,4 here and making a 1,4. I guess you're saying you take a 1,6 and do a 1,4. It's not the difference in the bonds per se. No other thoughts? Is that a hand I see or not? No. Don't look at me. Oh, come on, folks. Think about this. What else could there be? What drives, what determines whether reactions go or not? Energy, right? Okay. So maybe the 1,6 bond doesn't have as much energy as the 1,4 bond. There's not enough energy to drive that reaction if you clip it at the 1,6, and that's exactly the reason. If you go 1,4 to 1,4, the energy you gain in breaking this bond is used to put it on to this end. But there's not enough energy in a 1,6 bond to favor the conversion of that bond to a 1,4. That's why that happens. The clue is actually right here. Notice that we don't use phosphate to clip that off. In the other case, we could use a phosphate. There was enough energy in a 1,4 to use phosphate to break the bond. There's not even enough energy in a 1,6 to use phosphate to break the bond. We have to use water. Okay? So alpha 1,6 bonds don't have as much energy as an alpha 1,4 bond. Okay. Questions about that? The other thing I'll remind you, I mentioned it when I showed you glycogen structure before, is once we have a branch, then what we have is all of the bonds after that are 1,4. So here's 1,4, 1,4, 1,4, 1,4, 1,6, but then 1,4, 1,4, 1,4, 1,4, 1,4, 1,4. The branch is the only thing that's a 1,6 there. Yes, Jenny?
Why do we store glycogen instead of starch? This seems like a lot of work.
Why do we store glycogen instead of starch? She's asked the question that's going to lead me. I should have paid you to ask that question. So you're leading me to my next important topic. Okay? I'll answer that question directly. There's a very good reason we store glycogen instead of starch. Okay? And this is complicated because, you know, when you think about it, starch is very simple. You just go nip, 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 nipping, and you've got glucose. You don't have to worry about debranching, and when we go to make glycogen, we have to branch and all that sort of stuff. Why do we do this? Okay? Well, let's take a look at that. So if we look at, before I do that, let me get to this. Here's the phosphoglucomutase reaction. Glucose 1-phosphate, we put a second phosphate on it. We've got an intermediate glucose 1,6-bisphosphate, and we clip off the first phosphate, and we're now left with this guy. Okay? Let's think about glycogen structure. Okay? This is a schematic representation of glycogen structure. About every 10 residues, you'll see there's a branch, 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 branch. Okay? And glycogen structure is not at all unlike this in the cell. Jenny's question is, if we think about starch, or more specifically amylose, amylose, if you recall, is the component of starch that is not branched. It only has alpha 1,4 linkages. If we only have alpha 1,4 linkages, it makes breakdown and synthesis very simple. The problem arises from the nature of glycogen phosphorylase. Glycogen phosphorylase works beautifully on ends. The more ends that you have, the more glucose 1-phosphate that can be released very, very quickly. So you have plenty of glycogen phosphorylase present in the cell. The more branching you have, the more energy can be very, very quickly released. And this turns out to be very important for animals. Animals have to escape being chased. They have to be able to release a lot of glucose very quickly so they can get away from what's chasing them, or so they can go chase something, conversely. Either way, they need to have that sudden burst of energy. Plants don't have that need. Plants, unfortunately, don't get to run away from their prey. Okay? Not from their prey. You don't run away from your prey. You run away from... You know what I mean. You run away from what's chasing you. Okay? Your predator was what I meant. You don't get to run away from your predator, you're a plant. So plants don't have that need for a quick release of glucose from glycogen. So starch serves their purposes. You have a long, long starch molecule that only has one end, and you start breaking that down 1 or 2 glucoses at a time, you typically don't need a lot of glucose real quickly in a plant, and the plant is covered for that. So the reason is rooted in the nature of the way that the enzyme itself works. So this structure works very, very well for animals. And when you look across the animal kingdom, you will see glycogen abundant in muscle, and you'll see it also abundant in liver. That's the 2 most common places you will find glycogen. Okay, there's is the structure of glycogen. Glycogen phosphorylase. As you can see, it is a dimer, meaning it has 2 identical components. And it has an interesting coenzyme, okay, that is unusual in what it does. Okay, so I'm not going to talk specifically about what that component does here, but I want you to be aware of it. It's actually a vitamin component called PLP, and I'll say a little bit more about that. PLP is linked to the glycogen phosphorylase molecule through a lysine residue that you can see here, and not really much more to say about that. Okay. I know you guys would love to talk about mechanism, but I think I'll pass on mechanism. And now what I want to do is say a little bit about glycogen phosphorylase itself. So glycogen phosphorylase, what you've seen so far is you've seen how many enzymes involved in the breakdown of glycogen? 3. And 2 that were only directly involved. One was glycogen phosphorylase, which catalyzed the breakdown of glycogen. One was debranching enzyme that catalyzed the rearrangement of glycogen so that it could be further broken down. And the third was phosphoglucomutase, which allowed you to convert glucose 1-phosphate to glucose 6-phosphate. That's not even technically a glycogen enzyme. Okay? So now you know everything that there is to know about the enzymatic breakdown of glycogen, except, of course, for the regulation. And it's the regulation that I'm going to be talking about today, on Wednesday, and to some extent on Friday. Okay? So that tells you there's a lot of complexity to it. Glycogen phosphorylase turns out to exist in 2 forms. And we'll see that each of those 2 forms can exist in an R state and a T state. So in essence, we have 4 different forms of glycogen phosphorylase. 4 different forms of glycogen phosphorylase. That's a lot of different possibilities. Okay? Let's look at them. Okay? What you see on the screen is glycogen phosphorylase in the left in the R state, glycogen phosphorylase B in the right in the T state. What's the difference between A and B? Okay? So glycogen phosphorylase can exist in 2 different, covalently different states. And the covalent difference relates to phosphate. So if you put phosphates onto glycogen phosphorylase, you create glycogen phosphorylase A. If you take those phosphates off of glycogen phosphorylase, you create glycogen phosphorylase B. Okay? In my notes, you'll see I commonly refer to them as GPA and GPB. That's sort of my designation. A lot of other people don't do that. In fact, a lot of people will simply refer to the enzyme as phosphorylase. as if that's the only phosphorylase that's present in cells. Okay? It's a pretty important phosphorylase. All right, so the difference between these two guys relates to their phosphates. All right? As I said, each one can exist in an R state and a T state. So I can have an R state of glycogen phosphorylase A. I can have a T state of glycogen phosphorylase A. I can have an R state of glycogen phosphorylase B. And I can have a T state of glycogen phosphorylase B. Now, we're going to, you're going to sort of slip into this convention that I slip into and everybody else does and call glycogen phosphorylase A the active form. It's actually the more active form. And you're going to call glycogen phosphorylase B the non-active form, when in fact it's actually the less active form. Now what we're going to see as we look at this is that there are reasons why we think of glycogen phosphorylase A as the active form, and reasons why we think of glycogen phosphorylase B as the inactive form. That's not going to be apparent at first. Okay? But you'll slip into this designation as I do as well. Okay? Here, looking at glycogen phosphorylase, we see the different interconversions of these. So we see, for example, that in this case, B is on the left and A is on the right. B can exist in an R state and a T state. We see that phosphorylase A can exist in a T state and an R state. We see that either of these can be interconverted by the use of ATP to put phosphates on. What kind of an enzyme do you suppose is going to put phosphates onto glycogen phosphorylase? It's going to be a kinase. It's one of those rare times that it's not protein kinase A. It's actually another kinase called phosphorylase kinase. Okay? But suffice it to say that we can convert the R state of glycogen phosphorylase B into the R state of glycogen phosphorylase A by simply putting phosphate onto it. We can convert the R state of glycogen phosphorylase A back into B by removing the phosphate from it. What enzyme do you suppose will be involved in removing the phosphate? Phosphatase, okay? And you're right. So phosphoprotein phosphatase we'll say a lot more about on Wednesday and Friday. Phosphoprotein phosphatase is involved in taking that phosphate off. Now, look at the relative sizes of these directional arrows, all right? Notice that these guys are about equal in terms of going left or right, and that's because this is hormone-driven. If the hormones are present, it's going to go to the right. If the other hormones are present, it's going to go to the left. What hormones do you suppose favors the putting on of phosphate? Epinephrine, just like you saw in activating protein kinase A. It turns out protein kinase A actually activates the phosphorylase kinase. All right? So epinephrine now is going to influence glycogen breakdown. It's going to favor glycogen breakdown because it's going to favor the addition of phosphates to glycogen phosphorylase to make it glycogen phosphorylase A. On the other hand, insulin is going to work exactly the opposite. Insulin is going to favor the activation of the phosphatase, which is going to remove the phosphates. So depending upon whether we have epinephrine or insulin formed, we're going to have one or the other directions of these two, left or right. Well, how about the up and down? Okay. If we look at the up and down, we see there's a longer line pointing down to the T state of glycogen phosphorylase B. That tells us that given normal conditions in the cell, it's much more likely that glycogen phosphorylase B is going to be in the T state. Conversely, if we look at glycogen phosphorylase A, we see a longer line going up. That tells us that under normal conditions in the cell, glycogen phosphorylase A is more commonly going to be found in the R state. Now, it's important to note that they're not one-directional. There are times where glycogen phosphorylase A will be converted back to the T state. There will be times when glycogen phosphorylase B can be converted into the R state. That's the little short line going up. That's the little short line going down. I'm going to try to make you understand what those different conditions are. Okay, so before I go forward with some specific conditions with respect to oh, by the way, I should also point out, we're going left and right on this scheme. We're going covalent modification, either putting on phosphate or taking it off. As we're going up and down in this scheme, we're going allosteric control. So R state, T state changes always involve allosterism. They always involve allosterism. You don't make something R and T by putting phosphates on or taking phosphates off. Okay? Okay, so questions about this before I move forwards? You guys are asleep today. Too much turkey? You up for a joke? Yeah. I've got a dumb joke today, okay? You might say they're all dumb, right? How many people in here are pre-vet? Okay, so you guys will probably like this joke more than others. Maybe you've heard it, okay? So this lady, she walks into this veterinarian's office, and she's carrying what appears to be a dead schnauzer, okay? Hope there's no schnauzer fans in here, okay? And so she brings it in, she sets it down on the table. She looks at the vet and she says, "What's wrong with my dog?" The vet takes a look at it and he says, "Your dog's dead." And she says, No. She said, "It can't be dead." You guys like Monty Python and the parrot sketch? This is sort of the same thing. It can't be dead, right? It can't be. No, your dog is dead. Well, you've got to do something. You just can't tell me. I didn't come in here to have you tell me my dog's dead. He said, "Okay." He said, I can do something for you." She says, "Great." So he goes out and he grabs his little kitten, okay, brings it out there and puts the kitten in front of the dog, and the kitten comes up and sniffs it, and walks away. Lady kind of gives it the look like this, and he goes out and he's got this beautiful Labrador retriever, okay. He gets this Labrador and brings it in and does the same thing. Puts it up to the dog, and the lab comes up and sniffs it and walks away. He says, "Yep, ma'am." He says, "Your dog's dead." He says, "That'll be $700. And she says, $700? For this? Why $700? You told me earlier the dog was dead." He said, "Yeah, but that was before the CAT scan and the lab tests." I told you it was dumb. Did anybody not get that? Okay. All right, so let's spend some time now thinking about allosteric changes for glycogen phosphorylase. Okay, glycogen phosphorylase we're going to look at in the muscle. And by the way, I'm not overly concerned that we talk about muscle versus liver, et cetera. There are some differences, but we're not going to focus on those. It's much more important we understand what the allosteric changes are and how they work. Okay, this shows us, and it's unfortunately not showing you with the same proportional arrows that you saw before, so this is a little misleading. But this shows us glycogen phosphorylase B, in this case coming from the muscle. Okay? Glycogen phosphorylase B can be converted from the T state into the R state by AMP. Okay? It takes 2 AMPs because there are 2 subunits. And when that happens, when it binds AMP, it's converted into the R state. Conversely, glycogen phosphorylase B, when it binds to 2 glucose 6-phosphates, will convert back to the T state. Now, as I said when I started this lecture, there is a logic behind all of this. And I want to try to explain that logic to you, because if you understand the logic, the memorization really becomes a trivial thing. Glycogen phosphorylase B we think of as the less active form. So when would we want to have the less active form become active? Okay? I'll give you a real good situation. Let's say that you are sprinting, or you are scared and you're running away from something. Okay? Those 2 scenarios that I frequently describe to you. Okay? It takes a few seconds for epinephrine to get from your brain to the liver and target tissues to stimulate this process. Wouldn't it be nice if you could do this conversion so that you could start breaking down glycogen even before the hormone got there? Well, think about this. If you're sprinting, you're burning ATP very quickly. We've already talked about how creatine phosphate can help make more ATP, right? You remember that lecture in the muscles? But even there, you've got a limited capacity. You're going to start running out of ATP, especially if you're running really fast, really quickly. From the last lecture, I told you that you can take, when cells start running out of energy, what happens is they start accumulating ADP. And when they accumulate ADP, and they don't have a ready source of ATP, what do? They take those two ADPs, make one into an ATP and the other into an AMP. AMP, therefore, is a strong indicator of low energy, and look what it's doing. It's converting glycogen phosphorylase B immediately into the R state so it can start breaking down glycogen. That's really cool. Because glycogen is going to give glucose 1-phosphate, which is going to give energy to the cells and or the body that need it. In this case, it's working in muscle, and muscle is the tissue that's going to immediately need ATP. So we're not going to wait for the hormone to come and get us. We're going to actually have an indication of low energy turn on this enzyme. Because if I want to go from the B to the A, I'm going to need the hormone. And the hormone ain't here yet. Make sense? I don't have any other jokes. Okay? That was the one that was supposed to wake you up. Everybody stand up. Let's breathe deeply. Seriously, come on. Let's breathe, stretch. Okay? Everybody, jumping jacks. Get that turkey off. Okay? Come on. Come on. Does that help? Okay? You can stay like that if you want to. I don't care. People behind you may not like you, especially the people in the front row standing up. But yeah. Okay. Low energy is therefore immediately turning on this enzyme. The muscles really want that ATP all the time, because when the muscles run out of ATP, they're hosed, folks. They're not going to do anything for you. All right, let's go the other direction. When do we want to go the other direction? When the cell has plenty of energy, right? We don't want to be breaking down our glycogen when we have plenty of energy. You don't want to be lighting a fireplace when it's 90 degrees outside. You're going to contribute to global warming, and you're going to contribute an awful lot to your house warming. Okay? So, you don't want to be having glycogen phosphorylase very active when you have a lot of energy. Glucose 6-phosphate turns out to be a very good indicator of plenty of energy. Well, how does that work? Okay? Imagine, if you will, that we are going through either gluconeogenesis or we've plugged up glycolysis so that we can't break down glucose. What's going to start happening to intermediates? In both cases, they're going to accumulate, and they're going to accumulate around glucose 6-phosphate, because that's going to be the last thing before glucose. So glucose 6-phosphate turns out to be a fairly good indicator of the energy level of the cell. Plenty of glucose 6-phosphate is going to convert glycogen phosphorylase B back to the T state. The T state is going to be pretty darn inactive. It's not going to do very much at all. Okay? Now, I said earlier that we find glycogen phosphorylase B more commonly in the T state than the R state. What that should tell you is that we more commonly find glucose 6-phosphate present in cells than we find AMP present in cells. Only when we're really busting our tails do we have much AMP present in cells. Most of the time when we're sitting around, right now, I'll wager you that none of you have any AMP present in your cells at all, unless it's in those cells of your brain that are thinking very hard about what I'm saying. And I probably know better than that. Okay, that's glycogen phosphorylase B. Let's look at glycogen phosphorylase A, and we're going to look here in the liver cell. All right? Glycogen phosphorylase A turns out to be simpler. It's actually simpler. Because with glycogen phosphorylase A, you'll notice that in converting from the R state back to the T state, what converts it? Turns out glucose. What do liver cells do? What do liver cells make? They make glucose. Muscle cells don't make glucose, right? liver cells do make glucose. Ryan, you have a question?
Yeah, on the previous figure, it also showed that the CDR state had ATP. Does it also require ATP in addition to G6?
Let's see. Previous figure, right here. That figure? This one down here. Okay, glycogenphosphorylase B. Yeah. Oh, I'm sorry. I should have mentioned that. So ATP, of course, this is not a thank you for pointing that out. This is not a phosphorylation. This is an allosteric interaction. And so, again, ATP is an indicator of high energy. So, yeah, you're exactly right. I focused on this guy, and I should have said something about ATP. ATP, everybody knows, of course, is going to indicate the cell's got plenty of energy. It's got plenty of ATP. And it's going to flip the thing from the R back to the T. And that's not a covalent change. That's simply an allosteric interaction. Okay? Got that? Okay. Let's look at glycogen phosphorylase A and its forms of interconversion. So if we look at glycogen phosphorylase A, it's very simple. Liver cells are making glucose. When glucose starts accumulating, what does this mean? Well, let's think about this relative to the Cori cycle that we talked about on Wednesday. In the Cori cycle, what is the liver doing? It is taking in lactate. It is converting that lactate to pyruvate and then going through gluconeogenesis to make glucose so it can dump it back out in the bloodstream, right? When I'm exercising, that glucose is taken up by my muscle cells and used by muscle cells to make more pyruvate and lactate, right? When I stop exercising, the muscle cells stop taking up that glucose. So what happens to blood glucose concentration? It goes up. When the blood glucose concentration goes up, the liver says, no reason to put out more glucose. Liver glucose concentration goes up, and the liver has been told that you're shutting this guy down. Okay? So, glucose is allosterically converting glycogen phosphorylase A back into the T state. Yes, Robin? I'm sorry, I can't quite hear you. Yes. Is this an example of sequential? Okay, so her question is this an example of sequential allosteric interaction? And the answer is basically no, because in each case, and I think this one does as well, they each take two. So they're each both binding to both of those. So it's not, let me think about that. I can't tell you it's sequential versus concerted. Okay, I would argue that it's probably, I don't know now that I'm saying it. You could make a case for sequential. I'll look that up for you. I don't know the answer to it, but it's not a major consideration in how this overall process is occurring. Yes, sir.
Is this kind of like ATCA where it will
lock it in one? Yeah, it could work that way. And again, I just don't know off the top of my head. No. Okay, well, this covers going from the R state to the T state, all right? Look what it takes to go from the T state to the R state. Nothing. So what that means is when there's no glucose floating around the liver cell, glycogen phosphorylase A is going to exist in the R state. That's why we think of glycogen phosphorylase A as being much more active, because when it's present, it's usually in the R state. Only when glucose starts accumulating does it get converted to the T state. Now, I will remind you that allosteric interactions occur almost instantaneously. There's not a lag time for allosteric interactions to affect an enzyme. There's almost not a lag time. That gives the cell instantaneous control. That is very important because it takes hormones seconds to travel to their target destinations. Why might it be important to be able to control this very quickly? For the very simple reason that we don't want to break down any more glycogen than necessary. Okay? We want to be able to turn off this synthesis as quickly as the body no longer needs to be producing that glucose. We don't want to wait for the insulin to come around and favor the conversion of glycogen phosphorylase A back into the B. Makes sense? So it's complicated. You're going to see it actually gets a little bit more complicated when we overlay the hormones on it. But if you understand these basic principles of the interaction, the allosteric and the covalent modifications, then the hormones are going to make a lot more sense to you. So questions about these allosteric interactions or the covalent modifications? Laurie?
So is phosphorylase A predominate in the liver and phosphorylase B predominate in the muscle? Do they act?
Question, does phosphorylase A predominate in the liver and phosphorylase B predominate in the muscle? And the answer is no. A and B will predominate depending upon the hormonal state of the cell. That's the only thing that controls the amount of A and B. So if you have epinephrine, you're going to have an awful lot of conversion of B into A. And if you have insulin, you're going to have an awful lot of conversion of A into B. And that's going to depend upon the nutritional state of the organism and the energy state. But no, that doesn't mean that one is more abundant in one than the other. And those levels of each, B and A, are going to fluctuate a lot during the course of a day in any given cell. They're going to fluctuate a lot. Okay? Other questions about that? Okay, I've got just a couple of minutes. Let me see here if there's something I can do quickly. Otherwise, we'll stop early. Let's see. Okay, one quick thing. All right? Well, how do we convert glycogen phosphorylase B into A? We convert it using an enzyme called phosphorylase kinase. It's also called glycogen phosphorylase kinase. So you can call it either as far as I'm concerned. This shows what seems to be a complicated picture. Here's inactive phosphorylase kinase. Over here is fully active phosphorylase kinase. Well, how do we get from here over to here? It tells us what this stupid figure is trying to tell us. And believe me, I could have drawn a better figure than this, and I'm no artist or brain in terms of design. What this figure is trying to tell us is it takes two things for phosphorylase kinase to be fully active. One of these things is phosphorylation. And it's this enzyme that gets phosphorylated by protein kinase A. But you can see that phosphorylation only gets it partway there. It's only partly active. What gets it fully active is if we both phosphorylate it and we add calcium. If we just add calcium, we only get it partially active. Okay? So to get it fully active, we have to have both these things going on. Now, this turns out to be a really cool thing. Okay? The really cool thing is actually down here. I'm a muscle cell. What do muscle cells need for contraction? Calcium. Okay? They need calcium. Okay? As soon as the muscle cell starts contraction, it releases calcium. It immediately activates phosphorylase kinase in preparation for what it knows it's going to need more of soon, which is glucose. It's got a way to start breaking down glycogen at the very earliest instant of contraction. To wait for the full activation, we have to wait for the hormones to come and favor this addition of the phosphate. But at this point, even at this point right here, we have a phosphorylase kinase that is partly active. It's going to start phosphorylating glycogen phosphorylase B and making the A form so that we can start breaking down glycogen. Okay, that's a good place to stop. I'll say a little bit more about that at the beginning of the lecture next time.